There you go. All right. Plenty more prizes tonight. The greatest prize of all is, of course, we have Sergey talking again. Yeah. So Sergey only very recently led a talk, was it a few months ago, on collab design and hacking AlphaFold3, as well as other describing how other neural networks work for protein design and structure prediction. But now, the world changes quickly these days, and so now AlphaFold3 is out. The world doesn't know that much about AlphaFold3, and for various reasons. And Sergey is going to take us through what he can glean from the paper and from also using the server himself. I think he wants it to be something of a discussion. Is that right? So. His plan is for this to be something of a discussion. So. Like as always for these kinds of seminars, feel free to interrupt Sergey. Just don't heckle him too badly, but feel free to interrupt him with questions, and hopefully this will be an organic discussion about AlphaFold3 and how it's different and perhaps more enabling than AlphaFold2. With that, Sergey, take it away.
Oh, thank you very much. So today I'm going to tell you guys about AlphaFold3. In the past, we've always sort of done these sessions after CASP. Like usually after CASP was a new method that when we discuss it, no one really understands what's going on. And so then I could just say anything. But now there's actually a paper, so there's possibilities I'll say something incorrect. So feel free to correct me. So if anybody here actually read the paper more carefully than I have, you're welcome to be like, no, that's not right. And we'll continue the discussion that way. All right, okay, cool. So today, I'll sort of go through some of the background information. I won't go into too much background because I talked about AlphaFold2 before and all those details, so I kind of assume everybody here already is familiar with AlphaFold2. And so here the idea is more to tell you hey, what's changed since AlphaFold2? And what's different and what does the new model allows us to do? I'll describe some results. This is mostly just coming from the paper, showing 2 plots, I think. And then I'll give a quick tutorial for those of you who haven't tried using the web server. I think some of you guys are probably coming from companies, so you can't use it directly. But if you tell me what to do, I think I can do it. And then later on, you can just observe. I think there's nothing legal preventing you from observing. So this could be your chance to. And then towards the end, I'll talk about some limitations. So there's been actually a lot of experimentation, and people on Twitter have been posting lots of interesting observations. And I've collected a few that I thought were quite interesting. So I'll go through some of those as well. And then towards the end, if we actually have time, I'll describe how to invert matrices via power iteration. But that's maybe towards the end. There's a relationship to that, to everything else, but we'll see how far we can get. All right, so as you all know, AlphaFold3 came out. The paper's out, and that's really exciting. And so now to the background. Okay. So we know evolutionary information is very, very powerful. And what I'll try to show you guys today is it's still being used even in AlphaFold3. It's multiple sequence alignments that are passed into the model, and so it's very, very powerful. But those, just a quick crash course for those who now might be familiar, is when you have a single sequence, there's not too much you can say or do about it. Where things become really, really powerful, if you take that sequence and you search against a database of sequences, and then if you find other organisms that have the same protein, you assume they have the same structure and they have similar patterns of evolution, then you can start to imagine various constraints. So you collect a bunch of sequences of many different organisms, and this is where this information is really, really powerful. Like you get conservation from that, you can get that, and maybe you'll be able to say some positions are important because they're conserved, some positions are not important because maybe they're completely random. And this information is really, really important. But the most important thing which I'll tell you guys about today is coevolution. And so there's positions that are covariant with each other. And this is sort of like an idealized case. In reality, it's not always this perfect. Like you often have single mutations and then organism still survives, but then now you have a probability of maybe accepting the other mutation more likely than if that other mutation didn't occur. But once you have this kind of information, you can now use it to maybe infer structures. And so there's been a lot of work in this field about how do you use this convolution information to infer structures. And I do have a link here, so I won't go through all that background because I've done so in the past. But if you guys are curious, I do have a link here that describes all the various methods that try to use this kind of information. All right, so AlphaFold3, which I assume most of you guys are familiar with, we go, as I said, from single sequence, you build up a multiple sequence alignment, and then you pass it to a model like AlphaFold. The idea here, and I'll describe to you guys more in detail what each of these steps do, is you have a module that essentially extracts various constraints from the MSA, and then feeds those constraints to a structure module. And the structure module then tries to satisfy those constraints. And the really cool thing here is that if you actually start, the whole thing was trained end to end. So people have done sort of these individual steps before, but what made AlphaFold3 really unique was that it was trained completely end to end. And so there might be certain decisions that you make, like how do you extract coevolution? What sequences should you include, not include? How do you initialize the coordinates? How do you move the coordinates around? You essentially learn that all end to end. And that's what we believe is what made AlphaFold so successful. But one other thing that's really, really cool, which is something that hasn't been done before, is this idea to say, well, what if you sort of refine your predictions, right? So you make a prediction, you say, hey, actually, based on this existing prediction, maybe some clashes in the model, maybe the model doesn't make any sense, you can feed it back into the MSA module and sort of refine the constraints, and then essentially get new sets of constraints and try to using those new constraints, try to repredict the structure. And these iterations have been found to be very, very useful, especially in cases where you have large proteins or cases where the coevolution information is not very powerful. There's just not enough sequences. And so doing these iterations, you sort of improve the model. And so this is the details here. We won't go into too much details here, but essentially I'm using these neural networks as sort of just like cartoons, but in reality, each of these guys are like many, many layers, lots of cool stuff going on. And more specifically, we have the Evoformer, which is what I was referring to, the MSA module that processes the multiple sequence alignment, and then finally pass all this information through the structure module to the structure and you iterate. So it's the same image. And I just wanted to show this just to show you guys what I'm showing here is not completely made up, and that there's actually some connection to reality. Okay, so now looking a little bit more carefully at each of these modules. So now if we look at the MSA module, and oh, by the way, for those of you who are curious, like why am I showing you guys AlphaFold2? The reason I'm doing that is just to show you the contrast between the two models. And it turns out the model itself, AlphaFold3, is still very, very similar overall. It's just some of the details changed. So still getting a refresher what happened in AlphaFold2 is going to be very important to understand what's in AlphaFold3. So essentially you have an MSA module where you have the multiple sequence alignment. You go through a series of row attention, column attention. So you like compare every pairs of sequences, every pair of positions. And then there's one part which I will, if I have time at the end, I'll get into more detail here. There's this function called the outer product mean. And this is where essentially all the coevolution happens. You essentially compute a covariance matrix of all these features that come in, and then that gets updated to the pair features. So you explicitly consider every pair of positions and how they interact with each other and synthesize that information, add them to the pair features, and then you sort of iterate this process 48 times. And there's other cool things here like triangle attention. And those who are not familiar, this triangle attention is something we think is really, really powerful. What improved the coevolution extraction compared to previous methods. So previous methods have no information about what proteins are, any priors about proteins. But if we start to look at those triangle attention, what we believe these have the ability to learn is triangle inequality. So what I mean by that is if I tell you 3 sets of distances, like I tell you, let's say we have 3 points in the protein, like this little triangle I'm showing. There's this point, there's this point, there's this point, and I also have the distances between all those points. It's physically impossible for this line to be larger than the sum of the other 2 lines. And so these are the kind of things that the model can learn. And so, for example, let's say you extracted some interesting coevolution patterns from the MSA, and you're like, hey, it's physically not possible to satisfy all those distances. The model can learn to filter that information out. And so that's what we believe, like what the Evoformer, the main task of the Evoformer is to compute the coevolution and then essentially try to filter it through all these rules about things that we know about 3D physical space and triangles more specifically.
Sorry, I have a question.
Yep.
I have a microphone too.
Okay.
So my question is, did they in the AlphaFold 2 paper, they ablated a bunch of things and figured, this was important, this was not important for the success of the model. I can't remember, did they ever, delete the triangle attention and say this is how it affected the performance or not?
I think they may have actually, because they did try a few variations on this. One thing I do remember in AlphaFold 2 is a lot of these ablations they've done at the end. Okay, it got a little louder than I thought I would. Bring this down. Yeah, so it turns out on average, a lot of these things don't improve things that much, but there's always a few proteins where it improves a lot. So it's like if you look on average, it always feels like, oh, it's improving by like half a GDT or 0.1 GDT or something like that. But then it turns out like every protein, there's a few proteins where this helps a lot. And so like one example of this would be like, let's say if you have coevolution where there's contacts within the protein and between two copies of the protein, like a homoligomer. And so those are physically not possible to satisfy in a single structure. And so things like this could say, hey, actually these contacts, and start filtering those out. But for most proteins, that doesn't affect things. But it turns out there's at least, I guess I would estimate like 10% just to make up a number here, where that is important.
Okay, I mean, I'm asking because it seems like it would be important, and it is in AlphaFold 3. This so that's why I'm asking, like, if they ever actually deleted it and it's published that it affected things in some way. I just don't, because I did like 50 things in the supplement of AlphaFold 2 where they ablated things and basically nothing really mattered. But some things in when you grouped sets of ablations, it did matter. And I just don't remember the triangle attention stuff, if it was ever ablated, but maybe it was. So you're saying?
Okay, looks like there's a confirmation in the back that it was. Does anybody actually remember? Oh yeah.
I had it open, so yeah, it was, and it didn't have a huge difference. This is 2 GDT, and that's like 2 GDT for that. So it was like
Yeah, I think those ablation plots are sometimes slightly misleading because they're reporting the average across all the targets. But if you actually count the number of targets that improved over each of the ablations, you actually often see huge changes. Like it's like on average, everything's the same. But then if you were to plot like performance of one versus the other, you see like things above the line that appear. And so maybe another way the plot would have been probably better if they reported fractions of things that got better as opposed to the average mean. But thanks for checking. Oh yeah, another comment.
I had a weird question. Maybe you'll get to this. But,
oh yeah.
So me and my professor would always get in arguments of like, if you change the depth of the alignment, would that drastically change the quality of structures?
That's a good question. So I actually, in the previous talk, we've actually did this analysis. I'm gonna skip through a little bit into the past. So one thing we try to do is, as opposed to ablations, we just try, hey, what if we start removing information, right? And we did see, like, if you start removing information, that things get really, really bad. And what it'd be like, if you just give AlphaFold 2, in this case, just a single sequence, you see a drastic degradation in performance. So the x-axis TM score, and you can see a lot of proteins actually get really, really bad in prediction quality as soon as you remove the MSA. So it's definitely really important. But one other experiment you could do, which is kind of interesting, is to say, well, instead of just completely removing the MSA, what if I just like sort of shuffle all the columns of the MSA? Or let's say I only preserve the PSSM. But so the idea is that the other thing is that you could essentially collapse the MSA just to a profile. And interesting, the profile you see an improvement. So if you compare this orange population to this red population, you see an improvement overall when you do have this information. So here what we think is happening, and this is something we're not completely sure yet, is that the profile information actually helps in terms of maybe there are positions that are not necessarily co-evolving, but maybe have similar rates of mutation. And if they have similar rates of mutation, that might actually be an indication that they're interacting with each other. This is something that folks have seen in the past, I think back in the 90s with methods like mirror tree and trying to predict like which proteins interact with each other. And they saw that proteins that have similar rates of mutations tend to interact with each other. And so that's so those are kind of things that it might be picking up on top of just coevolution information. Yep. Okay. All right, back to reality. Oh, sorry, to the future, I mean. Okay, so triangle attention, right? So this is what we think is probably the main thing it's learning, is this idea that say, hey, every 3 pairs of distances, all 3 triplets of distances should sum up to a triangle. And if they don't, you could potentially imagine start to fix these things and iterate over and over and over. And then finally, once you've so essentially constructing these constraints, then you could pass these constraints to a structure module, and then the structure module essentially tries to satisfy those constraints. And so the interesting thing with AlphaFold 2 is everything initialized at 0, 0, 0, and then you essentially keep checking to see whether or not you match the constraints, and you iterate for 8 steps. And this works actually reasonably well, and actually works better than even initializing with an existing structure, which is what was done with a Rosetta Fold 1. And so you essentially just start at 0 and you unroll. And I like to think that this is sort of approximating MDS, multi-dimensional scaling, where you essentially have some distances and you try to move around the coordinates until they match those distances. But this approach, even though it seems to work, there was a few limitations of this approach. And the 2 main limitations of the approach was that, first off, it was deterministic, meaning that every time you give it the same constraints, it always returned the same solution. And so you can imagine a scenario where maybe the constraints have some ambiguity in them. Maybe you want to sample multiple conformations. And so people have been trying to hack this by sort of reinitializing, like essentially one way to fix the deterministic nature of structure module is to start messing with the MSA. Like you start to subsample the MSA, you put in some dropout layers, and then the structure module might be able to get through and give you a different solution. But the problem, I think this is one of the main issues, is that because the structure module is connected to the whole pipeline, like if you want to sort of sample a bunch of structures, you essentially have to mess with the MSA module. You have to mess with the inputs. Like, wouldn't it be great if you could just get the constraints and then just run this millions of times, right? So like, for example, the structure module literally takes milliseconds to run, while this MSA module, the Evoformer, is the most expensive part, and it could take hours to run depending on the size of your protein. So if you could sort of essentially stop here and then end here, you may be able to do a lot of cool stuff. And I think that's the main motivation of saying, hey, can we improve the structure module? So now jumping a little bit to the issues. So first off, there was no way of sampling alternatives without just rerunning the whole model from scratch. But then also the other problem that we have actually observed, some of the research we've been doing, is if you give it really, really large proteins, like even if you give it perfect information, like all the template information, it still gets stuck. It's not able to sort of realize a structure. So we think 8 steps is just either not enough, or maybe that's just not enough sampling. So it essentially becomes a sampling problem. So the solution is to move the recycling, just do the recycling in this part of the model, just cycle this, but then just have the diffusion just run once. But that being said, you could actually run diffusion many, many times independently of all the previous steps. So the biggest change, I guess I would argue, is that AlphaFold3 is this diffusion module, where now you can actually do those kind of things. And let me explain what those are step by step. So once again, here's a more detailed version. There's a lot of stuff going on here. I'm just showing this just to show you guys that I'm not completely making things up. And that you still have the MSA module, then you pass this to a pair former, which I'll explain to you guys in a second. You pass all these features to the diffusion module. You start with random noise, and then you keep updating that random noise to sample structure. But once you have the structure, then you could take that structure plus all the features from the previous sets of modules, and then try to assign some kind of score to that model, confidence to that model. And I think this part is really important because you could potentially run this many, many times and then use this part just for scoring or ranking. And so I think that was done intentionally because now you can run this thousands and thousands of times and then rank this with this part of the model. So just to summarize, we have lots of bits of information that's coming in here. Is we have the MSA, we have the templates, we have the conformations, specifically for if you're trying to predict where the ligand goes. That gets put into this embedder. The embedder embeds all this information. You get like single representations, pair representations, the MSA module, and so on. And don't worry if this doesn't make sense. I'll go through those in more details in the next few blocks. Okay, so what is diffusion? This is a nice slide that I've, I think, borrowed from somebody who borrowed from somebody else. But I think it was made by some person named Lou. So okay, what is diffusion? So it turns out what you could do is you could imagine taking an image and adding a bit of noise to it, and then training a model that undoes that noise, right? And you can just keep doing that iteratively over and over and over and over. And then the nice thing is once you've done this, you can start with complete random noise and just run in the reverse direction. Just undo the noise, undo the noise, undo the noise, undo the noise, and hopefully end with some cool looking pictures of a dog or a cat if you're a cat person. But the idea is like if you change the noise, you might actually coming up with a completely different image. And so this same procedure could actually be applied to proteins as well. So you can imagine having coordinates that you noise and then denoise, denoise, noise, and then noise and so on. And you just sort of learn models that add and remove the noise. I won't go into too much details how diffusion works because that's not my expertise, but there might be people here that understand diffusion better than I do. But the main key thing is that essentially the same exact procedure is now done inside AlphaFold3, where instead of starting at 0, 0, 0, like I said before, you start with random noise, and then for 20 steps, you just denoise those steps, one step at a time until you reach a structure. And I think it's still essentially learning something very, very similar to what the previous structure module is doing. But the difference here is that now you could actually start with random noise and maybe start getting different samples every single time you run, which is something to be wary of because if you change the seed, you'll get a completely different structure potentially. But it could also be a good indicator that there's enough information to realize the structure. Okay, before I keep going, any questions up to this point or concerns? Or yeah, I think there's a hand in the upper left corner.
Yeah, I'm just waiting for a mic or maybe.
Oh yeah, feel free to.
I'm gonna wait for a long time.
Okay, no mind. All right. Are there thoughts on the best way to construct this diffusion module? Like, are there better or worse sources of noise to add when you're working in a protein domain versus, say, images? I know there's some really strange work about adding completely bizarre noise structures to images, getting the same similar or sometimes better performance back. You kind of wonder how this might work with proteins.
That's a great question. I don't go into that much detail about this component, but one thing that actually was kind of interesting about AlphaFold3 is that they removed any kind of equivariance from the model.
Okay.
So what I mean by that is, I think for the last probably 4 years or so, people have been thinking like, hey, is there smarter ways of taking noise and denoising that noise that is somewhat respective of the directionality of the different parts of the molecule? And so one thing that's been interesting with AlphaFold3 is that they're like, hey, we don't have to worry about that at all, which I think probably made some people who study equivariant stuff a little upset because they're like, I think every paper they're like, oh, this is going to be important for the future versions of AlphaFold, but then they're like, oh yeah, it's not important. Well, that being said, they did do some tricks to try to get around that problem. So like one of the things that they did was they took the coordinates and they would randomly like permute them. By permute them, like they would rotate them, they would translate them. So the model may essentially learn some kind of equivariant function ultimately, but they didn't explicitly try to put that into the model. The other thing that's been shown in the image world is actually, it almost doesn't matter what kind of noise you do. Like people have shown that you can actually like pixelate, depixelate, you can make everything go to 0 and then go up. And essentially it turns out it almost doesn't matter how you noise or denoise. But that being said, I would assume for like more complicated topologies, you may have issues depending how you noise and denoise. Like, so one thing I'm not talking about today, but I talked about a little bit last time was these models like chroma versus RF diffusion. They do different kinds of noise. And because of that, they have different limitations. Like some are able to do better global sampling while others are kind of stuck just refining local features. So how you add the noise may actually play a role and for larger, more complex topologies. But in this case, it was like very, very simple, maybe a little too simple, which was kind of exciting because it's like, oh, it actually makes sense. Like just random Gaussian noise and then just denoising it and just learning denoisers. Mm-hmm. Okay, cool. Any other questions? Oh yeah. Or
just playing around, it seemed to, in some ways, hallucinate more than AlphaFold2, especially for like IDRs and stuff places where it shouldn't be structured. is that where it's coming from, or?
Well, that's a great question. So one thing that's kind of interesting about AlphaFold3, I guess, since we're doing journal club, let's see, we'll load up the paper. Actually, they recently updated the paper so that there's no longer like, oh, in preview, and some of the figures look a little nicer too. So if you want to revisit the paper with a more fresh perspective, but so one thing that's kind of interesting, and I think this is what you're alluding to, is this hallucination idea. So one thing that's been kind of interesting was, let's see, I think this was one of the figures here. Yeah, here it is. So one thing that we've previously saw is methods like AlphaFold3. Often, like if you're uncertain, you just sort of push everything away, right? It's like, I have no idea what's going on. And the reason why that actually happens is because there's an explicit function or loss function in AlphaFold2 that says move everything away. Like there's a clash loss. And one thing that actually happened in AlphaFold3 was they did not actually put a clash loss in. Why didn't it put it in? I'm not quite sure. I suspect it has to do something with how you train the diffusion module. Like you almost need to mess up the structure really bad in order to introduce some kind of clash loss. But if you're noising these only natural coordinates, maybe that's really hard to implement. And so instead what they did was they said, well, how do you get around this problem of just things getting packed? Is they actually trained on AlphaFold2 structures. So the diffusion module was essentially trained to noise and denoise AlphaFold2 structures, and that's how they were able to get around this problem. But there's this suspicion, and I don't know if this is completely true, this is just purely speculation, is that this ability to sort of noise and denoise these steps were probably restricted to a certain size or certain kinds of proteins. So one thing that's been shown already by other folks on Twitter is like, as you make the protein larger and larger and larger, suddenly it's like snaps into some new mode and then tries to collapse everything back into compact as much as possible. So I think it's a diffusion problem, and specifically because there was no clash loss added to the model, and that sort of creates this weird, how do you say, artifact. But as we know with AlphaFold2, one other thing that's kind of interesting is if you look at AlphaFold Multimer, you look at version 2.0, I believe, it also had the same problem. They didn't include a clash loss. And it was also packing everything and maybe hallucinating things to some extent. And then they tried to fix it, but it looks like they didn't completely were able to fix it with AlphaFold3. But that's something maybe they'll update in the future. So we'll see. I think if enough people complain, there might be a version 3.1 or something. Yep. Yeah, but that's a good point. So I will actually show a few examples of that later. And those who didn't hear the question is, the question is like, why does it hallucinate, or why is there hallucination going on in these diffusion models? Okay, all right. So so I described to you the diffusion, and I think that's all I'll say about that for now. But now, what's different about this other part of the model, this MSA and pair module? And I think this is actually the most important part, and I actually suspect that this is the main contributor, and that the diffusion part maybe is not actually doing as much as we are thinking it's doing. I think it's mostly because of the MSA and the pair module. And let me explain what I mean by that. So this here, I'm showing a picture of AlphaFold2 at the bottom. And so one of the interesting things in AlphaFold3 is they actually remove things as opposed to adding things. And what I mean by that, they remove the row attention, they remove the column attention, and then kept everything else the same. But more specifically, these 48 blocks were modified into 2 sets of blocks. And what I mean by that is there was one set of blocks that are with 4, and then a set of blocks that are 48. And the main thing, which might be a little hard to see here, is that in the first 4 blocks, you have the MSA, and that's being passed through the whole model. And then for the last 48 blocks, you're actually only dealing with the single representation. And so contrast that with AlphaFold2, is the MSA was dealt with all 48 blocks. And then with AlphaFold3, they're like, hey, actually, maybe we don't have to see the MSA for that many number of blocks. And I think this is actually quite important because this was actually the very, very slow step. Like passing the MSA 48 times was actually quite slow. Also, the row and triangle, sorry, the row and column attention was also quite slow. And so getting rid of that, and then just running triangle attention essentially on the same pair of features for 48 blocks was actually, did, I believe, 2 things. One is it made it really, really fast. So now, like, for example, if you use AlphaFold3 server, you can put in a protein of size 5,000, you'll get a result in 20 minutes. While with AlphaFold2, 5,000 residues would actually take maybe almost a day or two to get the results back. And so I think this is probably what contributed a lot. It's like you sort of remove the components that are not necessary. But the other thing is because now you have such a lightweight model, you're able to extend to much larger lengths. So one other big change from AlphaFold 2 to 3 was in 3, they actually increased the length to, I believe, 700 or something, while AlphaFold 2, I believe, was about 400 something in that range. If anybody here remembers the exact numbers, they can correct me. But I think this is what allowed them to do that, because technically with even like the best GPUs, like with 800 gigs of video RAM, it was impossible to fit a protein of length 5,000. And now by essentially getting rid of the MSA, only keeping the MSA in the first floor blocks, they were able to do a much better job. And so I think this is, and I think this is what made the model better, was just increasing the length. And so it's able to see potentially and deal with much larger problems. And it's usually the big proteins is where AlphaFold 2 struggled on. Okay.
They're still working with crops and proteins.
Yes, they're still working crops, but those crops are now the max of 786, something like that. I made up the last two numbers. Oh, yeah.
Could the difference in input size be actually the reason why that collapse or there was acting on larger complexes?
Yeah, that's.
That it gets distilled on proteins that can only be so big because they use AlphaFold 2.
I think you're right. I think you're right because at one point I think I was trying to do this test where I was like, I'm going to try to increase it and see when the collapse happens. But I forgot to do that. So maybe next presentation. But yeah, if I was to make a guess, I would say maybe around 800 is when the collapse will happen, because suddenly now it's never seen proteins that large, and sort of it's out of the distribution for the diffusion module. It's sort of seeing new pair features that it hasn't seen before. And what I mean by that is, like, if you imagine the pair features is just a square, and you're essentially seeing a bunch of things of I plus 700, it's like, okay, I've seen sort of this modality, and now you're beginning to see things out of distribution. And so what you start to do is you start to maybe hallucinate things. So I suspect that might have been the reason why. Yep.
So if you only need the full MSA for 4 blocks, and then the rest of it we just pairwise thing, does it mean that there's not much more information in the MSA beyond the pairwise?
I suspect, so I've done some tests, and I'll show you guys at the end of the presentation, is that I think 48 blocks with MSA is probably overkill. I don't think you need it for, my suspicion is maybe when they developed AlphaFold 2 is they just said, hey, let's just keep increasing the blocks and see what happens. And they kept seeing an improvement, but they probably didn't realize that each of those blocks were not necessary to have done all those things at every single part. It's like, it's probably turned out, my guess is given these results that they found, hey, you only need MSA for 4 blocks, and you've sort of already sucked out all the information you can from the MSA. And now let's just focus on refining those constraints. It's like, you don't really need to revisit the MSA more, because essentially at this point, you've already computed the covariance matrix 4 times. And there's no real reason to do that anymore. One little detail, which I will get to maybe at the end if we have time, is there's this pair weighted averaging. And I think this sort of takes essentially the steps of what the attention modules were doing, but a much more lightweight version of that, like not having to do full on attention and so on for each sequence. Okay, so now let me jump to the results. And so these are just results from the paper, but I'll also show some results from online. So the big thing was, if we compare AlphaFold 2.3 multimer to AlphaFold 3, we see for monomers, it actually didn't improve that much, right? So there seems to be just a little bit in improvement for version 2. Where things seem to make the biggest impact was actually for multimers. And so this is why I keep hitting back at large lengths. And my suspicion would be like, these few proteins where it got better is probably those much larger proteins. So the idea here is that maybe it's really, really hard to sample some of these really large conformations, but also just even deriving constraints for such large proteins. The protein antibody is a little bit, I don't know if the right word is sketchy, but it's not like they just ran AlphaFold 3, 1,000 random seeds, like completely running the model from scratch, not even just the diffusion part, just completely running the whole model from scratch. and then ranking all those solutions. And then but in the paper, they mentioned that even after 1,000, they still found that they can improve. They just kept sampling more. And so I guess I would say, to me, this kind of feels like cheating a little bit, because ultimately you should have a protocol that just gives you the solution in one go. But that being said, it also tells us that the problem is not completely solved. Like the sampling, like this diffusion part alone didn't solve the sampling problem. Oh yeah, sorry.
Do you mean it improves the PAE for multimers, or it improves like the ground truth answer?
Ground, sorry, I should have specified. So this is the percent of proteins that were successful. This is the success prediction, not necessarily the confidence. But the way they picked the answer was based on the confidence metric. So essentially you run it for thousands and thousands of times, and then you pick the solution. Sorry, just to step back a little bit to this big picture plot. You can imagine running this many times, just this part alone. But what they actually did was ran this whole thing many times with random seeds. And then sort of selecting. And so people in the field have shown that to be even the case for AlphaFold2. Like if you start to mess with MSA, you turn on the dropout, random seeds, you can sometimes get a much better solution. I think there's a guy, Jorn Wallner, I think that's his name. He has this method called AF sample, and essentially he just runs AlphaFold thousands and thousands of times and selects based on the confidence. And he actually found that he can actually pick out the correct solution based on the confidence. So it looks like, I mean, I guess my hope was that this diffusion step would have essentially solved or removed the need to do that, but it looks like it's still necessary, at least for antibodies.
Sergey?
Oh yeah, go ahead.
So I have a question. So when something is being passed from pair former to diffusion module, so what is it constraining the diffusion module in terms of protein geometries, right? So how does it constrain it to become from a noise to like a actual protein structure? Like in the case of RF diffusion, right? You're starting it from like a sequence independent random noise of coordinates to a protein like structure, right? So how are they passing the MSA pair former information where you have those coevolutionary constraints into the diffusion module such that they get a good structure?
I think I understood most of the question, but if I didn't, you might have to repeat this. But so the question was like, how is the diffusion module actually constrained? And so one big thing to keep in mind here is that methods like chroma or RF diffusion, they're completely unconditional. They're just generating things. There were instances where they trained on various conditions, but the big difference here is that this diffusion module, so the idea here is just to step back to maybe this little toy example. So here in this really toy example, you're starting with some image, you're noising and denoising, but imagine you also give essentially these arrows an extra label. So you say, hey, I'm going to predict the previous step condition, predict the previous step condition on the current step, but also condition on some additional bit of information, right? And so in this case, that extra conditioning is coming from the input sequence, it's coming from the pair features that come from the pair former, but it's also coming from, and also the single representation from that model. So this module is not just blindly denoising, it's denoising with those extra conditions. So you can imagine now when you're making updates or changing that random noise around, instead of randomly changing it, you're changing it to match maybe some kind of constraints. Overall, I like to think of these diffusion modules as essentially learning to do some kind of almost like gradient descent, given some kind of gradient. So you're computing gradients, applying those gradients over and over until you satisfy whatever gradients you have. But those gradients are conditioned on whatever input you give. Okay, I think yeah.
So the constraints are put in the objective function, or is it more in terms of features?
So the way the constraint, so the actual training procedure is kind of interesting. So let's scroll back. Yeah, here we go. So essentially what you do is, where's the steps here? Here's the step. So you have the ground truth, which you can also perturb as well, like you can move around. But
Mhm.
essentially you add the ground truth at the beginning, but you start adding various noise to that ground truth. And this is the training. But then what happens is because you have the network constraints, when you're learning to denoise the noise, you're conditioning it on the information that comes from the original model. So the constraints are added. Well, I mean, maybe when I'm saying constraints, that's more of a liberal way of saying it, maybe is like, it's not real constraints. You're sort of suggesting that this is the information, but the model we assume is learning how to extract those constraints and then satisfy those constraints.
Yeah, thanks.
Of course. All right. Anybody else have any questions? Oh yeah, go ahead.
So when thinking about the differences that you get in predictions when you rerun the diffusion module multiple times, when should we, I'm assuming most of us here, or users and designers, when should we be satisfied in our prediction quality? It's like I could run it, I mean, I only get 10 credits a day, but I can run it so many times.
Well, it turns out you get 20 now, but also you can make multiple accounts. And you can run it for as many accounts that you have access to.
So, but like, what, should we just use the quality metrics as a way of saying, okay, this is pretty
Yes.
good at this point?
Exactly. Yes.
Especially it seems like for thousands of times.
Yeah. So one of the unfortunate part is right now, when you run the AlphaFold server, it only runs one time. It runs one time, and I believe it makes actually 5 models, and I think those 5 models are coming from the sampling of this part of the protocol. But if you change the random seed, I'll show you guys later during the tutorial session, you could change the random seed and actually get a completely different solution. And you could actually run that multiple times and get alternate solutions, and then you could rank them based on the confidence. Yep.
So in previous AlphaFold, you said that the MSA part, I guess the entire thing is deterministic. So here, when they're rerunning the entire thing, are there parts of the MSA that aren't completely deterministic?
Right. So there's a lot of random things that could change. So when you change the random seed, if I understood correctly, somebody could feel free to correct me if I'm wrong. That still influences the embedder itself, like which sequences are sampled and used. And so when you change the random seed, you actually perturb the entire model, and that may result in slightly different constraints, which may result in slightly different structural prediction completely. So if, at the moment, unfortunately, the server does not allow you to input your own custom MSA. You can only use their own MSA, and that also prevents things from work. Like you can't. It's a little hard to do similar experiments that I described to you guys earlier. I think once you start getting the freedom to perturb the MSA, then that will be actually pretty powerful. You can start doing it. Yep. One little detail that I think I forgot to make a slide for, but one other interesting thing is when the MSAs are in cell constructed, usually if you have multiple proteins, you try to pair all those sequences together, right? And that's how it extracts co-evolution information. The one interesting thing about AlphaFold3 is that even the sequences that should not be paired are automatically paired anyways. And that seems kind of wrong, but I suspect what they did there was when they pair the sequences, they probably labeled them as paired. And when they didn't pair the sequences, they probably just labeled them as unpaired. And this creates kind of an interesting phenomena where before you essentially had a bunch of sequences that are kind of stacked this way, and you have lots of gaps. And I think that just created a lot of unnecessary wasted GPU time. And so by sort of essentially collapsing all of them and just labeling them, I think that also improved the results. I forgot to make a slide for that, but I think that's one thing to also keep in mind. I think that might have also played a role. Okay, so before we ran out of time, oh shoot. Okay, I should probably keep going fast. Okay, so far I've been telling you guys just about proteins and protein-protein interactions, but another aspect is adding support for small molecules, ligands, and RNA and DNA. I also wanted to put a plug in here for AlphaFold2 because that was also done there. And there's actually a lot of cool stuff here. And this is sort of two links here that you guys could check out. We'll post these slides later on the website so you guys can easily look at those links later yourselves. But essentially now you have the ability to specify more than just protein structure. And the results look quite promising. So this is just for protein ligand interaction. And so even for comparing to Rosetta Fold all atom, which is this method right here, it already appears to be doing significantly better. This is for predicting protein ligand interactions, but also comparing to some of the other methods in the past that essentially given a structure would try to predict where the ligand goes. They also do much better than that. So there's definitely something cool going on. I don't know if Nick has any things he wants to add here.
I'll just add that it's really hard to know exactly what this means without seeing what they trained on and what it is that, I mean, this was from the Pose Busters data set. It's hard to know exactly what 80%, success means when you can't compare against what exactly they train on. There's a lot of actually overlap, even in 2019 structures with what's in Pose Busters in terms of sequence identity and similar structures with ligands bound. So I would take these results with a sort of fairly big grain of salt.
So how do you actually run all this? So before I jump in how do you run this, I'll just quickly tell you guys about the limitations first. So there's essentially 20 jobs you can submit per day, but as I mentioned earlier, you can make extra counts. So technically, as many emails that you can create, that's the limitation. The other limitation is that there's only 5,000 amino acids. But one thing to keep in mind is 5,000 amino acids or 5,000 atoms, but 5,000 atoms for non-proteins. So for example, if you have a lipid, that will probably account for as many carbons as there are in the lipid. So this, like, for example, if you wanted to put a bunch of lipids in your protein, you could probably only fit about 300 lipids, and then you'll sort of run out of time. Finally, there is no code, which created a little bit of controversy because people are like, it's not cool. From this perspective of Nature, because I think Nature violated some of their own rules where they said that the reviewers should be able to see the code, but then apparently they ghosted the owner reviewer that demanded for the code. So there's a little bit of controversy, but the good news is DeepMind's, you know what, we're going to release the code in 6 months. So for those of you who are waiting for the code, it will come out in 6 months, but in the meantime, you guys can use the web server. The other limitation is if you're in it for the money, you may not be able to use the code. I think, yeah. But there is also one other thing. Martin Seneger brought up this point to me. He said, hey, actually, does that mean if somebody uses AlphaFold3 to solve some PDB structure, does that mean I can't deposit that coordinates anymore? Or if I do, nobody can use the PDB for commercial use anymore? So there's a little bit of a interesting discussion there to be had. Like, what does that mean in terms of academics then sharing the results? Like if they share it on the PDB, does that mean commercial people can't use it? So the cool thing is the code will be released in 6 months. And there's actually been multiple efforts now online of people trying to reproduce all the code. I think one example I wanted to highlight, there's this guy named Lucid Rain. I think his real name is Phil Wong. Actually, not just him, but other folks have been contributing as well on reproducing AlphaFold3. So at this point, it looks like all the code is reproduced. But the training itself I think there's been some groups that have been trying to train with this code, but the code is now essentially available. And besides Lucid Train, there's been other folks that contributed as well, like their modules. There's actually a pretty active Discord channel so if anybody here wants to contribute towards the code, maybe have some random GPUs laying around and want to contribute, I would highly suggest joining the Discord channel, which I think is somewhere in here. So there's a few efforts, and I believe like the AlphaFold folks are also trying to reproduce the code. So all is not lost for commercial folks. Okay, so because we're kind of out of time, I will kind of save the server because the server is pretty self-explanatory. It's like you paste the sequence, hit run. So I'll just quickly describe a few hacks that people have found with the server. And so one really exciting hack that I actually thought was really cool is you could actually just give it a bunch of lipids. And if you provide a bunch of lipids, it actually will place those lipids and start actually forming a bilayer. And if you have like transmembrane regions, they will actually start to interact with those transmembrane regions. I think somebody found that if you put too many lipids, you start forming micelles. And so another researcher found like, actually, if you mix the lipids, so it turns out there's 3 different kinds of lipids. So if you mix those 3 lipids together, you start forming bilayers again. So there's been kind of interesting hackery going on there to try to avoid this problem. One example that I'm actually working on, which I thought was kind of cool, was, so here's a protein and part of it is actually outside the cell and one part's inside the cell. And if you actually run it with AlphaFold2 or AlphaFold3, the intercellular and the extracellular part will come together. And so you get the wrong prediction. But it turns out if you just like start putting in some lipids, it actually starts to separate the inter and the extracellular part, which is kind of cool. So I tried like 15, 30, 60. If you go beyond that, it starts making micelles and then nothing works again. So there seems to be some kind of a critical mass where it starts to form micelles again. But it's actually kind of interesting. You could essentially so this helix used to be completely here, and then the inter and extracellular parts are coming together. And now you could essentially start to separate these things. Yep. I'm not sure actually. I don't know. I think it's learned like carbons like to be next to carbons. Yeah, because there are definitely lots of lipids in the PDB, but I don't remember if there's any actual bilayers in the PDB. Does anybody know? Okay, well, so that's one cool thing. The other interesting thing, this is something Tomek has realized was, so if you take a full-length protein, like a large protein, and you start to truncate that protein, AlphaFold3 will still return say super confident about this truncation. Like it's exposing hydrophobics on the surface, and AlphaFold3 still thinks this protein is fine. But one thing that I found that was kind of cool is if you start throwing salt in there, like literally putting a bunch of Na and Cl atoms in there, now suddenly this truncated protein is less confident. So I think it seems like you can use essentially salt as a way to tell the model, hey, this is not a unit in isolation. Like my guess is here, it's like saying, hey, this is a truncated protein, and it's probably implicitly assuming there's other things around it. But as soon as you throw salt in there, they're like, hey, say this is a bad idea because you're exposing lots of hydrophobics on the surface, and the salt can interact. The one thing that's unfortunate is that they don't allow you to put in water. I think if they let you put water, then that would have been better, right? You could have just flooded everything with water, and then it should have said, hey, solvation is important, right? So right now we can't have salt. We can't have water, but we can have salt. That's one exciting thing. Another example that I thought was kind of fun that got this protein structure, this alternative structure actually came under different conditions. Like there's actually, they discussed salt in here. They said, hey, if you increase the salt, you get this conformation. If you decrease the salt, you get this conformation. If you increase the temperature, decrease the temperature, you get different conformations. And so in reality, if you actually just throw a bunch of salt in there, you actually get the structure that is in the literature reported as this conformation with lots of salt. Unfortunately, there's not a really good way to increase or change the temperature, but I think once we get access to the code, we could probably increase the temperature in the diffusion module. Right? You could say, let's add more random noise in there, and that's, I guess, one form of temperature.
So was AlphaFold3 trained to also predict where water goes? Or did they strip water out and just did ions and ligands?
It's a great question. So I don't actually remember.
I don't think so. From the paper, they didn't mention that.
Trying to remember exactly. Oh, boy, this is a random protein. I try to predict a bunch of salt, lipids, and 2 alanine helices. So it's you can that's what the world looks like from AlphaFold's point of view. So like in this case, kind of makes sense. You have these are all hydrophobic, right? And so you're packing all the carbons against the all the helices, and then you have to put salt all around. So looks like it's working. All right, so these are some positive results. Another example, this is actually kind of fun because it's Nick Blitz's design. So Nick's like, hey, there's all these molecules I can't put inside the protein. Like, how do I, okay, one thing I forgot to mention is another limitation of the server is that you only have access to a certain kinds of ligands. So when you click entity, sorry, let me zoom in a little bit. And then you can pick, for example, ligands. But when you pick ligands, you actually have a limitation to only ligands that are essentially natural. So things that are typically found in natural. I think there's two potential reasons for this. I think one hypothesis is that that essentially only works well on natural ligands. But the second hypothesis is they're a drug company, right? Isomorphic. They probably don't want you to make money off of this until they do first.
That's the same question for water, right?
Mhm.
Can it deal with water? If it can deal with water, the reason to not allow it is because it's actually quite powerful.
True, exactly. So one thing, if we go to ions, I was actually kind of curious, like, oh, maybe I could make water with hydrogen and oxygen, right? But turns out they did not put hydrogen or oxygen as one of the options. So but this is something that would be fun to experiment with once the code comes out, or if LucidTrain re-implements or retrains the model. So there's been actually kind of interest. So one hack say, you know what? Why don't I just put every possible ligand in there, right? You just list all of them, and then just see, do any of them land near the active site? And it turns out, in this case, some of the molecules did actually land where one of these NIC's ligands binds. So it could potentially be used as a way to sort of screen for binding sites. So even if you do, and then maybe if you have, you know what your ligand looks like, maybe it looks something like one of the natural ligands, you probably hack your way through. Okay, and I won't go into too much detail, but there's a guy named Luciano. He's actually went through Twitter, has already combed all the cool results, and he put them in this blog post. So you guys could check out his link. He actually went through a lot of cool examples beyond the ones that I've shown today. So if you just plug in here. Okay, now for the last session, I'll go into warnings. So the first warning is, as one of the folks here already alluded to, is hallucination. So this guy named Jacket. Okay, sorry. I forgot. So this person named Jacket, he realized that, hey, if you give this protein to AlphaFold3, it creates all these nice looking spaghetti. If you're Italian, you probably would appreciate that more. But it turns out if you put in another protein, suddenly all that spaghetti just becomes all ordered. And so then the question is like, well, what's happening? Does this protein become ordered upon binding this? But in reality, what happens is if you just have a huge protein, it just even like, so sorry, let me rephrase this. So this right here is the same as this picture here. And so here it's disordered and it's not confident at all. These are PAE matrices showing the predicted alignment errors. And so for the regions that are not ordered, it's putting them down as spaghetti, which is what we expect. And also it's not very confident. But when you start to move to more and more larger context, it starts to order all those spaghetti together. And so one thing to be cautious of is if you just look at this, I think before people used to use spaghetti as a rule of thumb, is this confident or not confident? Like you get an AlphaFold prediction, you're like, hey, if it's spaghetti, it's not confident. But in reality, you could still get non-spaghetti for non-confident regions. So it's really important to continue looking at the confidence metrics because that things may still appear to be well predicted and well packed, even if they're not confident at all. So that's one warning. Another warning is if sometimes you put multiple copies of the same protein, instead of forming a homoligomer, the proteins will actually start to overlap on each other. And so that's been reported by multiple people. And in this case, I believe this guy named Kevin, which I think he might be online here. So he feel free to interrupt if I'm saying this wrong. But he noticed there were some clashes upon introducing higher order information to AlphaFold3. So it looks like it has maybe some symmetry breaking problems where if you have too many of the same protein, it just doesn't know how to sort of separate or break them apart. So which kind of would make sense in diffusion. If you have things pulling, everybody's giving equal forces, you kind of don't know where to go. You can get stuck. So there's still problems like that.
I have a question.
Oh yeah, go ahead.
So if we don't have experimental data, how can we be sure that there is wrong structure or real structure that might be existing in the work?
So in this particular example, like I would say this is wrong just because there's literally two proteins superimposed on each other. So I guess we would assume it's wrong, but who knows? Maybe there's an alternative universe out there where everything is possible. But that's a great question. But on a more serious note, with AlphaFold 2, people have shown that you can actually start increasing the number of copies and score all the different confirmations of different number of copies. It's just one of the problems with AlphaFold 3 is that when it's not sure, it's just sometimes just overlaps the copies on each other. And this is actually something we've also noticed in AlphaFold 2 before. Like if you take AlphaFold 2 and you disable the recycles, you also see the copies just literally overlapping on each other. So my suspicion, just to come back to what I think is happening with AlphaFold 3, is with AlphaFold 2, you actually you made an attempt of a prediction, and there might be clashes in that prediction, right? And once you see those clashes, you actually feed those clashes back into the model, refine the constraint, and try again. But one thing that happened in AlphaFold 3 is they removed that recycling step. Essentially, there's no longer any feedback from the structure back to the Evoformer itself. And I think that's the reason why this is occurring, is because there's now no longer a way for the model to sort of correct itself. It's like it made an attempt to embed, but there's no effort to tell the model back that you've screwed up really badly. And I think part of the reason why this is happening is because if you think about it, it's actually hard to propagate through all diffusion steps. Like in reality, the way people train diffusion step is like essentially picking a different step to pass gradients through during training. And so if you want to feed the model back in and then pass all the gradients through everything, and I think that's what prevents you from using diffusion models for that purpose. Like you can't propagate all the gradients through all the iterations of the diffusion. So that's my hypothesis why we see this kind of thing where molecules are literally overlapping on each other. Another example, and this is something Christine and Claire shared with me actually today. I hid the protein just in case it was insensitive, but this is actually a really exciting example where this is a protein and a nanobody docked to it. And initially when they approached me, they're like, hey, with one antibody sequence, AlphaFold is really, really confident. Another antibody sequence is not confident. Like, what's going on? And I said, well, maybe you should just increase the number of seeds. And so what they found was in this particular example that when they did like 20 seeds, 1 out of 20 seeds was really, really confident. So here, it might be a little hard to see here, but these are predicted alignment error matrices. So here is, there's 2 copies of this protein, and there's 2 copies of the nanobody. And so you can see it's really, really confident how those 2 proteins come together. Nanobodies are confident, but they're not confident how those 2 are docked together. And so if you do seed number 5, you do seed number 9, you do seed number, what is it? Seed 9, none of those work. And then it turns out the magic number for this example was seed 14. So this to me suggests that I think it's very similar to that antibody example that I told you guys about where they had to run it for 1,000 times. And this is particularly the exact same example. There's no coevolution information between the antigen antibody, that's an antibody. And so you need to do lots of sampling to find the solution. And so in this case, Claire had to do 20 times, which is just enough for that day. And there was one seed that happened to work. I suppose she could have stopped at seed 14, but it's a little hard to predict if that will be the case. Yeah, so one thing to keep in mind is that if you're thinking of using AlphaFold 3 as some kind of screening, it may be possible that it's still a valid protein and valid interaction. It's just maybe that one seed was just not lucky. In fact, like every time you run AlphaFold, so like in this case, not sure why that says preaches, but or preaching. But so essentially by default, it just creates some random seed. Like every time you run it, it just randomly generates a number. But if you actually click on seed, you can actually specify the seed by hand. Looks like the smallest seed is 1. So essentially you can iterate these seeds and then submit the jobs 20 times. There is a way to, like, let's say if you're a little don't want to do these things by hand, there's actually an option to upload a JSON file. So you can actually create a file, and there's actually pretty good documentation here where you can essentially create a file with lots of random seeds. Like you could specify the seeds and it will actually automatically submit all those jobs with all those seeds for you. So if you get to a place where AlphaFold's not giving you a good prediction, you just keep running. Highlight for benchmarking. So if you guys want to see additional benchmarking case where it worked, where it didn't work, they've actually did a pretty good job on that. The other really cool thing is this guy named Simon recently did a dive into the AlphaFold architecture and tried to sort of explain what AlphaFold 3 does for non-ML people. He actually has a really cool blog post, which actually just came out today. I actually asked Simon if he would be willing to travel to Boston today and present instead of me, but he said he was unable to do that. but he does go through in great detail about every single step of what each part of AlphaFold3 is doing, and actually has a nice breakdown of every single step. And I think it ends with an actual blog post that probably lots of cool stuff. So definitely check it out. I think I recognize that image from somewhere. Yeah. Also, I think Carlos did something similar too. So, all right. So bonus. So one of the things that, for me personally, what I'm curious about is like, where is the coevolution actually computed? And I think I already alluded that it's computed in this outer product mean. But what I want to do is sort of investigate this a little bit more carefully, try to understand, is that where it's actually computed, and is 4 blocks even enough to actually compute the covariance matrix or coevolution matrix? Just a little bit of background. How do we actually compute coevolution? This is the diagram I showed before. The way we usually do it is we have a multiple sequence alignment, and imagine we have a very simple scenario where there's 2 positions covariant with each other, and this position is conserved, right? And so one very, very simple model you can apply to this data to extract these covariance models, the coevolution signal, is through a linear layer. And what I mean by that is, imagine you have a sequence, you can one-hot encode that sequence. You could pass it through a very, very dense, very, very simple dense layer. And that's where all the coevolution signal is stored. And then you could try to minimize the difference between the input and the output. And you can imagine if you actually do this, your model won't learn anything useful. It will just learn to pass all the signal through, right? And so one trick you could do, and this is something that was done in the method direct coupling analysis, DCA, is to actually add some regularization. So for example, you can regularize the trace of the model. You can also add some L2 regularization to promote sparsity. And what you end up getting is this very, very simple analytical solution called the inverse covariance matrix. So it turns out if you just minimize the difference between the input and the output, you don't actually have to run this numerically. You could actually you can get an analytical solution of this problem by just simply taking the inverse of the covariance matrix. There's a few other factors in here. And so, for example, you would add some kind of shrinkage term based on the L2 regularization. If you guys are curious, I have so we have some papers here that you could take a look at that goes into more of the math, exactly how this was derived. But the key here is that there's an inverse covariance that needs to be computed. And so then the big question for us becomes, so just to step back a little bit, is this somehow computing the covariance and then taking the inverse? Because that's what we know is important in order to extract accurate coevolution signal, right? Okay, so now if we extract, so for example, once we fit this very, very simple model, we can extract the weights of this model into this little tensor, and then we can reduce this tensor and get a contact map. And so this is what we've previously used to predict structures. And so the question is AlphaFold3 doing the same thing? Is it somehow doing all this procedure for us? So this slide is a little busy, but you're welcome to ignore it for now, or I could walk you through it. But essentially, if you think about like what exactly does it mean to take the inverse covariance matrix? So here I'm just showing a little bit of math here, but maybe in a more Pythonic way. So imagine you have your variable x, which is your multiple sequence alignment. You can compute a covariance matrix, and then you can decompose that covariance matrix with methods like SVD or eigendecomposition into eigenvalues and eigenvectors. Now, you could then also resynthesize your exact covariance matrix by just taking the outer dot product of the vectors multiplied by the values. But the interesting thing is to take the inverse, you literally just divide by the values instead of multiply by the values. So the inverse matrix is just taking the covariance matrix and dividing it essentially by the values themselves. Now, the reason why this is kind of interesting is because where does this large eigenvalues come from? And they're usually coming from these indirect correlations, right? So you might have positions that are coupled with everything. And so now if you divide by that largest eigenvector, that's essentially across the whole matrix, you're essentially downweight those indirect correlations. And so this was the big DCA breakthrough was, so if you guys are curious, you could read more about here, is this ability to get direct correlations as opposed to indirect correlations. Now, the reason why this is kind of cool is because the question is, how do you actually compute eigendecomposition? So this is getting into a bit of a rabbit hole here. But turns out you can approximate eigendecomposition, or in fact, actually exactly compute it via power iteration. And then what is power iteration? It turns out power iteration is essentially taking any random matrix that you want to decompose. If you just multiply it by a random matrix, you could actually approximate the largest eigenvectors. And so you could essentially keep doing that and keep approximating, subtracting them, and you may eventually arrive at the inverse. And so it turns out, and so the reason why I'm telling you all this is because essentially inside AlphaFold3, you have the outer dot product, which is essentially computing the covariance matrix. And then you have some random math going on here, which going through it to me, it appears that it's possible to actually derive the power iteration within this this MSA pair weighted averaging with gating. And don't worry, it's not going to get any more intense than this, but essentially I try to simulate it. So I said, okay, let's try to actually do this, right? So let's say we start with some random representation, which is our MSA. We compute the pair matrix, gets a random matrix, random weights, which I'm just going to call params. I'm going to go ahead and keep subtracting them over and over and see at what point will I arrive at the inverse, right? And it turns out you could get there in about 20 steps or so. So what this here is showing is the number of these random iterations. Here I'm also showing the animation itself, where the slide 0 or frame 0 is essentially the covariance matrix. And if I just keep running this very, very simple math here, and if I just keep doing that over and over, I can eventually arrive at the inverse. And I think the part that's really cool here is that I can get there maybe in 10 steps or so. And this is like completely random. There's like no learned parameters here. This is just literally taking random parameters and just using random parameters, you can essentially invert the matrix in 10 steps or so. Yep.
Is that independent of matrix size?
Yep. Oh yeah, it is. Sorry, let me rephrase that. It's independent matrix size, but it is dependent on whether or not your matrix has dominant eigenvectors. So for example, if all the eigenvalues are the same, essentially if you just keep doing this over and over, it's not going to change your matrix much. But this is actually the cool thing is that the problem is, like, let's say if you just keep removing random, say approximations like eigenvectors from your matrix, eventually you'll get to a point where random does not change your matrix anymore because you've now removed all the dominant components and now you arrived at the inverse. So the idea is here is you just keep doing this over and over, and eventually you're now all the eigenvectors are essentially identical. Sorry, all the values are identical, and so now you're inverted everything. And so the reason why I think this is actually really, really cool is because all the components of doing what I just told you are all present in these two blocks of this model, like this outer product mean and this MSA pair weighted average with gating. So like you have the actual covariance computed here. You're also here, you're adding, like you're taking the previous pair, so previous MSA, you're adding the new from here, the new MSA, you just keep doing that. And so it turns out all the parts are there. So I guess what I'm just trying to say in conclusion is that I think you did it. Yeah, but anyways, that's all I had for today. Well, thank you very much, you guys. We still have